Good afternoon and welcome to today's webinar on long axial field of view and total body PET imaging. Today's session focuses on an industry perspective of total body PET imaging. My name is Dr. William Drew and I'm the PET Engagement and Delivery Manager for the UK's National Total Body PET Imaging Platform, NPIP. Long axial field of view PET imaging represents one of the most transformative advances in medical imaging in recent years, with dramatically increased sensitivity, improved quantitative performance, reduced administered doses, and the ability to acquire dynamic imaging data across the whole body simultaneously. These systems are opening new opportunities across both clinical research and drug development. As the technology continues to mature, long axial field of view PET is increasingly attracting the attention of the pharmaceutical industry, where there is growing interest in how advanced imaging approaches can support translational science, improve decision-making in clinical trials, and accelerate the development of precision medicines. Imaging has long played a critical role in drug development, from understanding disease biology and patient stratification through to assessing therapeutic response and supporting regulatory submissions. The enhanced capabilities offered by total body PET now provide opportunities to generate richer biological data, improve study efficiency, and potentially answer scientific questions that were previously difficult or impractical to address. NPIP has been established to help ensure that the UK plays a leading role in this rapidly evolving field by enabling collaborative research, supporting clinical translation, and developing the infrastructure and partnerships needed to realise the full potential of next-generation PET imaging technologies. We're therefore delighted to be joined today by two highly experienced leaders from the pharmaceutical industry who are actively shaping the application of advanced imaging technologies within clinical development. Dr. Phil Murphy from Johnson & Johnson and Dr. Natasha Patel from GSK. Dr. Phil Murphy is Head of Imaging and Digital Health at Johnson & Johnson within the Immunology organisation. He holds a PhD in clinical MRI from the Institute of Cancer Research and has spent more than 25 years working across the pharmaceutical industry, including roles at Pfizer, GSK, and Johnson & Johnson. His career has focused on advancing innovative imaging approaches within clinical drug development, spanning translational science through to late-phase, multi-centre clinical trials. We're also very pleased to welcome Dr. Natasha Patel, Oncology imaging scientist within the Oncology clinical development team at GSK. Natasha has a multidisciplinary background spanning chemistry, molecular imaging, and translational oncology research. She completed her PhD at King's College London, focusing on molecular imaging of antibody conjugates in preclinical oncology models, using PET imaging and mass spectrometry imaging techniques. Her work has included immune cell tracking using zirconium-89 PET, and exploring how advanced imaging approaches can support early clinical development and disease characterization. We're very pleased to have you join us today, and we look forward to what promises to be an engaging and insightful discussion. Before I hand over to Natasha and Phil, I'd like to remind you to please submit any questions that you may have into the Q&A box on your screen, and we'll aim to address as many of these questions as possible following the presentations. So I'd like to hand over to Natasha and Phil.
Perfect. Hi, everyone. I'm Natasha, and today I'll be talking alongside Phil about the role of PET and long axis field of view PET imaging in immunology and oncology drug development. Just before I start, I just want to mention that the content of this presentation and any other commentary made during this talk are reflective of our own personal opinions and not of our employers. Next slide, please. So this is the sort of structure that we'll talk through today. So starting with the landscape of drug development in 2026, and then we'll move into oncology, followed by immunology, and at the end, we'll discuss future directions for long axis field of view PET. So to kick things off, I'll hand over back to Phil.
Great, thanks so much, Natasha, and good afternoon, everyone. It's a pleasure to be here. So we're going to kick off this session talking about some of the current themes in drug development today as we start to figure out what the role of imaging is, the role of PET, and then the role really of the next generation of technology that we're seeing play out. So to set up some of the themes that we're seeing in drug development, here are sort of 4 areas where I think are telling us about the direction of drug development today. So if we start on the left, the therapeutic platforms, modalities that we use for targeting are becoming more complex and broader. We have everything from small molecules to antibodies, everything in between, cells as therapies, gene therapies, a lot more complexity in terms of the therapeutic platforms. Obviously, imaging has an important role as we try to figure out how those platforms interact with tissue. Translational science now is really fundamental to the way we work and really often built into most pharma organizations, and really thinking about it from an integrated measurement point of view. We can use tissue, blood biomarkers, imaging, and increasingly using those things together. Critical interface between the discovery organizations and the early clinical space. Precision medicine really developing from the roots in terms of targeting, matching drug to patient in oncology through to figuring out how we do that in other areas. It's a field that's growing pretty fast. And I think imaging certainly has a role to really understand stratification approaches. And AI, of course, is really ingrained in a lot of what we do. Opportunities, of course, from an imaging perspective, particularly when trying to translate complex data sets through to decisional endpoints that we need in pharma. We go back to a slide many of you have seen and many of you are very familiar with drug development, but just to sort of set some of the directions that we see in this field. So we of course step through a series of phases of increasing complexity and scale through clinical development from phase 1 to phase 3 towards a regulatory review. And relatively few of these candidate molecules make their way through to receive an approval. This has been the way typically for a long time. So we have a lot of failure across the industry. These take a long time to get there, and it costs a lot of money to do so. So these are some of the challenges that we work through, and I want to focus a little bit more on those. The other thing to note is as we step through towards later in development, these are just example costs. They vary across different areas, but they really step up in terms of cost. When we want to fail, we need to fail early, and that's a theme that I want to follow through on today. When I started my career all those years ago at Pfizer, actually, I think I remember looking at some of the statistics on success rates of drug development, and it was really surprising to me. I couldn't quite think of an industry where you had only a 10% success rate of a candidate starting in the clinic and getting right through to successful approval. So it was a challenging space, and it's been interesting to sort of follow how that's changed over time. It's a number I've kept my eye on for 20 years, and you probably may be surprised to know that it's actually gone down. The success rate now can be measured a number of different ways, but it is standing at about 5%. So in 20 years, what have we done? Well, I think it's fair to say that there are increasing number of approvals if we look at this over a number of decades. Certainly an increasing level of R&D expenditure over that time, and you can sort of see those two areas sort of diverging, those two plots are diverging. But it's not all to be despondent about. I think what we are certainly seeing is a tremendous amount of innovation. So complexity is certainly increasing, more complex trial designs, perhaps into new diseases that we haven't been able to consider before, evolving regulatory standards. So it's become harder to do. Complexity has certainly increased. But to address that, certainly also in step with that, R&D spend has increased too. All those new therapeutic platforms are now shaping small companies to large companies, and the level of ambition is shifting from, in some diseases, from control to remission through to even talking about cure. What we really have to do now is focus on the success of our clinical programs, improving those success rates. And I think the failures that we see are not because of a lack of innovation. It's the opposite. There's a tremendous amount of innovation, but perhaps it's knowledge gaps. It's really understanding our medicines very early so we can predict success later on. So we can accelerate medicines that have got promise and stop those early. that don't look as though they have the potential. So that's what we're going to focus on today. If you think about what really drives failure, a lot of it is clinical efficacy through these reasons you can see here. And really to set up the discussion on PET, I think these areas on the right are really fundamental components by which we can apply next generation PET to really help us across a range of diseases, deeper understanding of the diseases that we study, understanding mechanisms in the right tissues, and identifying those early compounds that are efficacious versus and accelerating those versus failing faster, those that don't have the efficacy. Understanding patient heterogeneity, a really fundamental opportunity, I think, for next generation PET, and also thinking about how we can do faster, smarter, and even trials with relatively few subjects. So I think each of those areas quite generic, but I think there's a lot of opportunity for next generation PET technology with these approaches. And with that, we're going to go into a couple of areas now. Natasha's going to take us through oncology, and then I'll jump back in to take us through immunology.
Yep, great. Thank you, Phil. Next slide. So, as many of us are aware, oncology remains as one of the most active areas of global R&D. And this slide notes that in 2024, there were over 2,000 oncology trial starts, and that novel therapeutics, including cellular and gene therapies, ADCs, bispecific or multi-specific antibodies, represent a substantial proportion of oncology trials. And this is important because these treatments increase the need for mechanistic measurement and understanding. So, for example, in ADC, we care not only whether the antigen is present, but whether expression is sufficiently homogeneous across disease sites, whether the drug penetrates lesions, and whether payload delivery translates into biological effect. Next slide, please. So, as I just mentioned, the drug development pipeline is more complex and more biologically rich than ever. But the main bottlenecks are still patient stratification, target engagement, spatial tumor heterogeneity, and early decision making. And these are areas that PET could add value. It's also important to note that the science has sort of shifted as well from single target settings towards more heterogeneous tumor biology, acquired dynamic resistance, and more complex tumor microenvironments, and also the use of combination therapies, which all in all makes conventional tissue biomarkers and single lesion biopsies slightly less representative of the full picture. And as we know, biomarkers now guide several stages of oncology drug development. And this is from diagnosis, prognosis, to patient selection, response monitoring, and even assessing resistance. And imaging is a key component of this. So PET can really help us understand and visualize more about target engagement and early pharmacodynamic signal. And from a pharma perspective, this is where PET becomes more valuable. So not as an extra scan, but as a decision-making tool that can improve patient selection or support dose and mechanism questions, and ultimately help us make earlier and more confident go or no-go decisions. Next slide, please. So now if we sort of hone into imaging a bit more now and what's currently used today. So the current state is that it's still dominated by structural assessment. So CT and MRI are central for things like eligibility, baseline tumor burden, longitudinal assessment, and resist response. And then we have two separate sort of categories when it comes to PET. We have established and emerging PET. So FDG PET is well established for things like staging, restaging, and response. But we also now have emerging target-defined PET applications, such as PSMA, somatostatin receptor imaging, HER2 imaging, and others. But in clinical trials, PET is often seen more as supplementary rather than decision driving. And this is because RECIST is, it remains deeply embedded because it's standardized, it's very familiar, and it's also operationally feasible. Whereas, in contrast, PET-based tumor response frameworks, they do exist, such as PERSIST or Lugano, but broader adoption of more novel PET endpoints has been slower. And this is interesting because it's not because PET lacks the biological value, but it's more because new imaging biomarkers need that standardization and clear context of use that they improve decisions beyond existing tools. Next slide, please. So PET imaging can help address critical gaps in oncology by sort of bridging this divide between anatomical structure and cellular function. Specifically, first with whole body target mapping. So instead of relying on one biopsy, PET shows target expression across all measurable disease sites. In terms of patient selection, PET can help identify patients whose disease actually expresses the relevant target in vivo, and then importantly, whether enough of the disease burden expresses it to justify treatment. Third, for PK/PD and target engagement, um, we can get serial information on biodistribution and tissue penetration, and also some early pharmacodynamic effects that we can pull out. And this can be very powerful in things like dose selection, especially when plasma PK does not, accurately reflect tissue pharmacology in certain drugs. And then in terms of theranostics and dosimetry, when when the diagnostic ligand and the therapeutic ligand are biologically aligned, PET becomes more than a biomarker. It becomes more of a treatment paradigm. And this is something I'll touch upon in a later slide. And then when considering long-axis field of view PET, that technology can potentially strengthen each of these gaps by enabling dynamic whole body kinetics, lower dose, and improved sensitivity. Next slide, please. So this slide gives a select few examples of the different roles that PET has played thus far. So just to pick out a few, so FDG PET in Hodgkin lymphoma is a classic example of PET being used more for treatment response assessment. But then we have things like PSMA PET in prostate cancer, which illustrates patient stratification and companion diagnostic style selection. So all in all, these examples show that it shows two things. Firstly, that PET succeeded in oncology where the clinical question was simpler and validated, and also actionable. Um, but some of these specific examples also show that the newer generation of oncology PET tracers has shown biological promise, but the broader clinical uptake has still been relatively limited. So next slide, please. So just to dive into a couple of these cases in more detail and looking a bit more at the newer generation probes. So one of the clearest success stories is PSMA PET in prostate cancer. And here, imaging is not just descriptive or visual, but really used to select patients for therapy. And even the FDA states that patients for Pluvicto should be selected using an approved PSMA PET product based on tumor PSMA expression. And this example worked because the PET readout is directly linked to the therapy mechanism, and the clinical question is sort of go or no go, as well as the workflow being embedded in a theranostic treatment pathway. The second example is HER2 immunoPET with zirconium-89 trastuzumab. And this example is very scientifically strong, although not as widespread in the clinic. So HER2 immunoPET demonstrates the appeal of PET for whole body target heterogeneity imaging, but it hasn't still become routine because antibody-based PET is still a little bit operationally difficult, slower. It still lacks the level of standardization needed. And there are also these longer imaging windows that need to be taken into consideration for patients. And the last example is FAP PET. And so FAP PET has been very promising, but it's also an example that has not yet fully worked through. So it's exciting because it targets the tumor microenvironment, and in certain cancers, it can outperform FDG, especially in less FDG-avid disease. But it also illustrates the broader challenge that despite the enthusiasm and the results that have been seen so far, applications are not yet in, clinical guidelines or clinical use extensively. And there's still this limitation that interpretation can be confounded by benign fibrotic or inflammatory processes. Next slide, please. So there are undoubtedly several practical barriers to scaling novel PET in oncology. So one of the main ones is manufacturing and capacity are major constraints. Some specialist tracers do require the radiochemistry infrastructure and reliable supply chains. Standardization is another major challenge. So for multi-center trials, we need harmonized acquisition, reconstruction, quantification, and analysis, because without that, it becomes difficult to interpret longitudinal or cross-site data with confidence. And sort of a related but sometimes underappreciated barrier is also site feasibility. So it's not just whether these PET scanners exist, it's whether the right patients can access those scanners, whether the site has experience with the relevant tracer, whether imaging slots can be coordinated with the protocol window, and whether clinical trial investigators are engaged enough to integrate the imaging endpoint into patient flow. And alongside this, there are also, of course, regulatory and cost considerations, which are important. You know, a novel PET biomarker needs evidence of added value. We really need to show that it improves decision making or patient outcomes in a defined context. And finally, there is still biological complexity. So a single target tracer still cannot capture the full tumor ecosystem. Especially when you consider things like resistance, immune contexture, stromal biology, and, heterogeneity, all evolving over time. Um, so yeah, overall, the barriers are not just simply technical, they're scientific, operational, regulatory, economic, and also biological. Next slide, please. So yeah, all in all, I believe that the future is not that PET can be used everywhere, but the future is more that PET can be used where it answers a question that other tools cannot answer as well. And the next sort of wave could be led by theranostic pairs, whole body target expression imaging, early PK/PD imaging in phase 1 and 2, and also long axis field of view PET. And to sort of summarize this section, for PET to earn its place in drug development, it really needs to be tied to a clear biological target or a therapy decision or prospective context of use, or also evidence that simpler biomarkers cannot answer the same question. And so the last point is important from an industry perspective. So PET is quite resource intensive, so it needs to be deployed strategically. But when it changes who gets treated or what dose is selected or whether a program continues, or even how response is interpreted, it can be extremely valuable. So yeah, with that, I'll hand over to you, Phil, to talk about the immunology context.
Great, thank you so much, Natasha. So, immunology area really contrasts to oncology. I think in oncology, you're really building upon a base of how it's used both clinically and in trials in certain settings for many years. Immunology is a bit of a wide open field. I think there's a lot to do. It's really an area of growth from doing very little to, I think, a lot of opportunity, and really keen on to hear your thoughts on where we could take this. But I think it's a tremendous potential. I'm going to start just by outlining immune-mediated diseases for those that don't follow this area. So think of diseases of the joints, rheumatoid arthritis, for example, inflammatory bowel disease, lung diseases with an inflammatory drive, skin diseases, a whole range of different diseases that define immune-mediated diseases. They're very heterogeneous. There are many of them. They're very complex from an immune system point of view. Complex systemic biology covers the whole body. They're very dynamic as well, differences from patient to patient, differences over time within the same patient. And I purposely chose a slightly unusual example to present in a PET talk, which is a skin disease, just to really drive home the point that in clinical trials, for example, in atopic dermatitis, we typically measure the skin lesions, and we can measure how they respond over time to treatment. But it really doesn't give us that insight into the full immune manifestation. If you look at the right, these are examples of things that are altered based on this particular disease. Even functional brain imaging can show differences in patients with these skin diseases. Aortic inflammation that we know drives increased cardiovascular risk, even shared biology with asthma and other diseases. And also shared barrier dysfunction, shared immune dysregulation with inflammatory bowel disease even. So although it's a skin disease, it really is about how it manifests in the whole body. And that really encapsulates the challenge of studying immune-mediated diseases, is that they're really, really broad in terms of how they interact with organ systems. So how do we typically measure them? I measure these through clinical development, measure these diseases and the therapies we have coming through. It's typically broken up into two areas. I think of translational science, thinking of that bridge from discovery to early clinical, and then we hand over to clinical development to take it through, define efficacy in larger patient populations. We use a range of different measures to do that. So in translational science, it's all about the mechanism, understanding the biodistribution, target engagement. For that, we can use tissue. If we can access tissue, we can use blood for peripheral biomarkers if they're going to be useful. So we use a range of different tools really focused on the biology. And then we move towards clinical development, and it's much more towards outcomes. We do structural assessments. We ask the patients through patient-reported outcomes of the symptoms improving, really focused in a very different way. We're kind of having a way, this sort of blunt transition, if you like, between the two. And I really think PET has this opportunity. It's really taking whole body biology and being able to take us towards clinical development, studying mechanistic response in the whole patient, understanding stratification based on the whole body biology and not just a peripheral marker. But it's also about how we complement with these approaches, measures that we can do in blood and tissue. We also contrast these approaches, and it really, we really think about where we can get tissue, a small sample of tissue. We can do really exquisite measurements to understand cell populations, for example. So it can be very information rich. But clearly, that very localized sampling is not necessarily going to reflect the full heterogeneity of how all organ systems are involved. It's unlikely to. If we go to the other extreme, blood, very high frequency, straightforward to do. But those measurements may not reflect what's going on in tissue. And I think, again, molecular imaging sits in the middle. It allows us for those whole body assays, even if there are some gaps that we currently have in terms of the biological interpretation of it. We'll talk a little bit more about that. But I think it's really important to see all of these ways of assessing the immune system together. It's not about selecting one versus the other. And to build on that theme of how molecular imaging can bridge between the early biology insights and the outcomes on the right-hand side. But I use this example of rheumatoid arthritis, and this is such a nice example of an image from UC Davis from this paper, and these images were presented a few years before that at a conference, and I think it just gave fantastic sort of insights into how you can look at disease. This is with FPG, but just with fantastic resolution, very high sensitivity, looking at a disease in a whole body context. And I think it really does step up in terms of giving us these insights into how the immune system behaves when we can look at all tissues together. But importantly, how do we then relate that to these other measurements? Well, it can bridge to the biology. We can compare it to tissue and circulating biomarkers to understand those measurements further. But also we can connect to things like structure and therefore outcomes. So I think molecular imaging here, particularly with the step up that we've seen, can give us that much important bridge between translational science and what we do then in clinical development. I have to, with this slide, sort of shout out to the field and what's been done in the last decade or so of pulling through an incredible range of traces that are immune cell specific or associated processes, a real array, a toolbox, if you like, of different approaches with tremendous amount of work, the expertise of the biologists, the radiochemists, the clinical expertise to guide these towards clinical evaluation. So this creates a fantastic tool set, I think, to really build upon the opportunities with the measurement and next generation PET, and perhaps there are gaps in terms of our understanding of the biology that they're reporting on. Perhaps there are gaps in terms of specific targets, but it's a phenomenal base showing what can be done, which will, I think, really elevate the opportunity for imaging the immune system for years to come. And I look at a couple of examples, and again, I chose FAP again, like Natasha pointed out. It's a really interesting tracer. These are standard PET scans, but I wanted to make 2 points with these really nice papers that we've seen. The one on the left is really driving home this point that if we want to look at inflammatory fibroblasts in rheumatoid arthritis, these are 4 patients. The paper gives examples of many more patients, but what just strikes you is the heterogeneity. And it's really hard to understand disease in any other way when joints are affected to different degrees. You know, we've been studying RA with X-rays and MRI for many, many years, in fact, decades. But this just gives such different insights into the biology at the whole body level. So really capturing heterogeneity. High sensitivity measurements will take this even further. On the right-hand side, a really nice paper diving in depth at the immune profiling of tissues alongside FAP PET, showing that in treatment with anti-TNFs and anti-IL-17s, you can see these response. So it's a movable endpoint as well. And on the right-hand side, just starting to show those structural correlations with molecular imaging. So I think these types of examples are really taking us down a fantastic track and really gives us reason to believe that these techniques could really move along what we do in immune-mediated diseases and developing new drugs. This is the slide that I sort of always say keeps me up at night. These are the challenges of when we start to look at the immune system with PET. I've been involved in immunology organizations for about 15 years now, and I'm sort of an amateur immunologist. I learn every day. The one thing I do know, it's very complex. Populations, subpopulations, dynamics, overlapping markers, spatial distribution, changing over time. It's really, really hard biology. So can PETs start to help with some of the gaps that we see in understanding the immune system? We know the challenges with PET. They're well defined. I think the key thing is not trying to compare PETs to the exquisite measurements we can take from a very small piece of tissue. We're not going to be able to compete with that. It's a very different measurement. I think with the right tracer at the whole body level, it really does give us unique insights. So again, using tissue alongside the whole body measurements is really, really critical. So in terms of the trajectory, particularly thinking about next generation PETs to study autoimmune diseases, there is no doubt this is unique information that we're seeing, the true whole body measurements of immune mapping. Natasha mentioned low dose protocols, and particularly for autoimmune diseases, immune-mediated diseases, it's really going to take us into patient populations that we've been hesitant to study before, where we want to do multiple scan time points, if we wanted to use long-lived radioisotopes, for example. It really does open the opportunity to study different patient populations. couple of other areas in 3 that I think are really important: the right tracers, and our understanding of existing tracers, but also the analytics, the measurements, can be really interpreting those incredibly complex whole body data sets towards decisional endpoints that we can use in drug development. So I think the opportunity is there now for immunology, and there are many layers of innovation, I think, to take us even further. And with that, I will pass on to Natasha as we sort of wrap up.
Yeah, thanks, Phil. So now in these last few slides, let's sort of bring the oncology and immunology together and focus a bit on long-axis field of view PET. So next slide, please. So this may be knowledge that many of you already know, but this slide captures the main technical advantages of long-axis field of view PET. So one of the first major advantages is the sensitivity. So it can provide substantially higher sensitivity than conventional PET, which enables us to detect lower abundance biological signals and also quantify activity in smaller tissue volumes and, as mentioned before, work with lower injected activities. And sort of that becomes particularly important when we're imaging subtle immune biology or low density targets, such as small lesions, or wanting to look at early pharmacodynamic changes. The second advantage is reduced radiation exposure. So, from a drug development perspective, that's important because, firstly, it can broaden the populations we can study, but also makes serial imaging more feasible and safer for patients in longitudinal studies. The third and perhaps most transformative advantage is dynamic whole body imaging. So examining tracer kinetics across the entire body simultaneously, that opens the door to whole body kinetic modeling or organ level biodistribution, and extracting a bit more quantitative PK or PD analyses. So for oncology, this could support tumor heterogeneity assessment, whole body drug biodistribution, and provide early pharmacodynamic readouts, and more so in safety-focused evaluation of off-target tissue uptake. So in that sense, long-axis field of view PET expands PET beyond staging and response assessment into a bit more mechanistic and decision-oriented applications. And then for immunology, the value is equally compelling. Long-axis PET could enable systemic immune mapping, multi-organ inflammatory burden assessment, and safer repeated imaging in chronic immune-mediated diseases. Next slide, please. And sort of to summarize, drug development does need better biology-driven decision tools. High failure rates reflect, at least in part, incomplete understanding of the biology, and this is where imaging can play a greater role. PET is already proven in some contexts, particularly in oncology, but the adoption, as we've seen, is quite variable. In oncology, PET is pretty embedded clinically in some instances, but it's still underused for mechanism and pharmacology in trials. But in immunology, PET still remains more underutilized despite the more systemic nature of many immune-mediated diseases. So long-axis PET represents a step change because it enables this whole body assessment. But the impact, it will depend on integration, not just this neat technology alone. We need collaboration across biology, imaging science, clinical development, translational medicine, and so on. And in addition to this, we also need strong academia industry partnerships because no single group can solve the tracer validation, infrastructure, and implementation challenges alone. Next slide.
Great. So I'm just going to wrap up with this last slide, and then we'll shift over to questions, and really looking forward to thoughts. I think these are some actions really we're thinking about from an industry perspective in terms of how we can catalyze adoption, as you've heard. And I think there's so much opportunity here. Oncology has used these tools for many years and arguably underused PET, but what do next generation measurements, where do they take us? For immunology, as I said, wide open opportunity. I'm really thinking the value cases. I mentioned that example of the rheumatoid arthritis example. It's really examples like that where you start to think about disease differently that really makes a difference. And convincing those that are not familiar with PETs that actually this is a technique that can really inform our drug pipelines. I think what's important also thinking from a radio tracer point of view, that we're thinking about what pharma are working on in terms of early pipelines, what's going to be relevant in 5 years, 10 years time. Let's try and figure out what tracer gaps there are at the moment that we can work on, particularly across industry, whether with pre-competitive opportunities. We've talked a few times about some of the barriers to adoption. Those are important too. In some cases, when we go into brand new diseases like skin disease, for example, it's awareness because dermatologists, I can tell you, are not used to using PET. So these are very new approaches for many new clinical areas, but with the approaches, we're gonna head into these new diseases. It may be evidence technical, and we've heard about some of the operational challenges too. I think a couple of things around the analysis. I think this is really important. So what we do in pharma is we move to clear and as crisp as possible decisional endpoints. And so often when we use PETs in certain settings, certainly in oncology and some other areas, we kind of distill down incredibly complex data sets to a single measurement. It could be SUV max of the most active lesion, for example. So how are we going to do that? I don't know the answer to that, and hopefully someone on the call and the wider field will help us with this. How do we shift whole body, very complex data sets that really capture the complexity of disease? How do we translate that into decisional endpoints that we can use in drug development? I think it's a really interesting innovation challenge, sits alongside the radio tracer challenge, but there's plenty to do, but I really think there's plenty of opportunity if we can share the right challenges to work on together. And that's me done. Thank you.
Thank you, Natasha. Thank you, Phil. So we have a stream of questions that are coming into the Q&A box. I'd like you to continue to submit these. We'll try and address as many as we can. We'll just work our way through them one by one. Phil, just touching on that last point that you made, do you think there's opportunities, for new machine learning or AI techniques to kind of do that distillation of, really complex, really large, complex dynamic data sets, for example, from a total body PET image scan, to lead that down to those decision-making endpoint data that you described?
Yeah, I think there has to be, and I'll definitely defer to the experts, of which there are many on the analysis frameworks for PET and those really thinking about this field, because I know it's rapidly changing. I suppose as we develop more, data sets in PET, that will be a really important force to help us develop the right methods. I think it's probably worth a focus area at some point, because if you think about those FAP examples or the FDG example in rheumatoid arthritis, you've got 30 or 40 areas of uptake. That there has to be a better way of some sort of cumulative measurement of the immune insult, if you like, or something like that. So I think we have to think a little bit differently, and I'm sure AI and machine learning methods will absolutely be part of that. And I'll defer to experts to guide us on which approaches we should be taking. But I'm very happy to give the challenges as we head in that direction.
Thanks. So we'll work through the questions from top to bottom. So we have a couple from Heather Williams from the Christie Hospital in Manchester. So the first one relates to funding. So yeah, obviously, it's always a difficult subject to address. But Heather comments that they've received comprehensive funding for trial setup would improve participation together with harmonized uptake for novel isotopes that can be applied across multiple trials. And she gives an example of an FCG PET core lab trial. At the Christie, they've received funding for camera and calibrator calibration for zirconium-89 and Environment Agency site license modifications for 225 actinium And Heather's wondering whether there's any possibility for pharma to potentially support that as a standard. So I don't know if that's something you have a comment on.
I, I mean, I totally support that idea, and we have to do. I guess from the studies I've been involved in over the years, I've seen many PET studies go wrong because of quality, because we haven't put the effort in up front to standardize for the calibration approaches. So every patient counts, and if we're going to shift these to decisional endpoints, we have to put that work in. So I just say, Heather, absolutely agree with you. We have to find mechanisms to do that. Whether we do that within a study and pre-work, or we figure out collaborative networks to support that as well with pharma funding from other places. I think there are different ways to do that, but wholly supportive.
Thanks, Phil. I think the next question relates to immune markers as well. So maybe this one's for you to comment on. So again, from Heather. So radiopharmaceutical availability is also a challenge. Do you have a view of the number of immune markers that are currently available within the UK? Heather comments that they've used the 89 zirconium CD8 marker and or PET tracer. And Heather's also aware of some limited studies using TSPO and FAPI.
Yeah, I think there's a list. I think in the UK it's relatively small, and I think there's work to be done to be able to address that. I think we probably need a discussion on this to figure out what, and this isn't just, I'm not talking about just what J&J wants, but all companies generally, where is the pull? Is it CD8? We want to look at macrophages, B cells. There is a list, neutrophils. What should that be, and therefore what should we prioritize being available for our trials? So I think there's work to do in terms of the UK positioning itself to access these traces, but I think we also need to figure out from an industry point of view, how do we prioritize the right probes to be available? So it's a bit of both ways around. Very happy to engage on that topic.
I had a question actually. When you put up your chocolate box of immunology tracers, I wondered if you had one in mind where there's an area where you'd like the academic radio pharmacy community on the call today, where people were looking for new opportunities, if you have a suggestion where they could explore developing new tracers.
There are always many. There's a shopping list always. I mean, some great things we're working on that already that others have contributed to and made available. The fibroblasts are a very exciting story with FAPI. Let's see where that goes. I think traces for neutrophils could be really important, very relevant to a range of diseases. B cells as well. There's been some work done, but I think B cells are definitely going to come back in need. And then things like barrier biology as well, relatively untapped area, very relevant for the gut, and other area, and the lung as well. So there's a list. Again, maybe we can find a way to get together across industry and put that list together and figure out a way of pulling some of these things through. Again, we can't solve these for any program. I think the most value is in traces that have broad applicability. And so we have to have pre-competitive approaches to solve some of this stuff. I just totally think that's the only way we can address it. And multiplexing as well. If there's a solution to that, it's a challenge. So not go there today.
Thanks, Phil. Maybe we'll come to Natasha with the next question. So the next question from Phil Blower from King's. So we've talked about oncology and immunology perspective in this presentation. Phil was wondering if you had any comments on cardiology applications.
Yeah, okay, thanks, Phil. I mean, I don't have a huge focus on cardiology, but I can say that it is also a very compelling and adjacent area for long-axis field of view PET and PET in general. Because cardiovascular disease is often systemic and inflammatory, metabolic, and quite dynamic. So there is a strong use case for whole body PET, but also quantitative PET beyond perfusion potentially. It can look at assessment for things like vascular inflammation or myocardial inflammation, and even potentially sort of detecting off-target cardiac toxicity. So, I mean, traces such as sodium fluoride, FAPI, they could be especially relevant and try and interrogate metabolism or fibroblast activation, looking at FAPI, or sort of microcalcification and things like that. So, yeah, I mean, from a pharma perspective, I think the value is translational and probably safety focused. It could really help quantify sort of multi-organ cardiovascular risk, for instance, or monitor inflammatory pathways and things like that. Um, so yeah, there are definitely cardiology applications. But yeah, I'm not an expert in it, so it's not something that I've dived into a lot.
Thanks, Natasha. I'd like to draw everyone's attention. I think it was the third webinar in the NPIP webinar series was led by Professor David Newby from Edinburgh University, and that had a cardiology focus. So maybe that's something you could look up on the NPIP website. So Phil's other question is relating to the industry's view on how the shift from conventional to long-axis field of view PET will impact the design of diagnostic tracers, such as the choice of targets and also radionuclides. Don't know if anyone has a view on.
It's really interesting. So I guess from an industry perspective, there are two dimensions to it. I mean, so those industries that are developing diagnostic agents, I mean, I would have thought that's an obvious route into well characterize with high precision new drug diagnostic candidates. I guess from their perspective, then how do you scale that? You have to then think about a broader network with standard PET to do it. I guess from our point of view, in terms of is a little bit different. I guess from in terms of an industry position on labeling assets, where we can answer questions in small studies. I think we absolutely have to use this PET. I think it's the only way to keep radiation dose and measurement quality at a point where we can use the data. So I guess there's just a difference between the diagnostic pharma world and our world in terms of where we take this.
Yeah, and can I just also add on, potentially the choice when it comes to choice of traces and things, I guess the radionuclide choice could potentially broaden as well because, we have that higher sensitivity which can support lower injected activity and shorter scan times and things like that. So, I mean, the radionuclide still needs to match the biological half-life, but yeah, it could be potentially more useful for long-lived isotopes, such as copper-64 or zirconium-89. Yeah, but I think in general, I think it still probably doesn't remove the core development requirements for a tracer, for instance. I think industry will still prioritize targets with a strong disease relevance where there's a clear context of use and scalable manufacturing and things like that. So yeah.
Thanks, Natasha. Terry Jones has asked, so Phil, in your final slide, you touched a little bit on some of the sort of challenges to adoption. However, Terry's asked around how to consolidate the collaboration between academic PET and drug development in pharma.
Yeah. No, that's a great question. Thanks, Terry. I think we see it's interesting. So on the pharma side, particularly those that haven't been engaged in PET, for example, immune-mediated diseases, where many clinicians, biologists are not thinking about PET, there's a whole gap. There's a whole load of folk that've got a lot of expertise that haven't thought about PET and what they want PET to do. So that's kind of a real untapped opportunity. I think what we probably have to do is create a really refreshed interface. I think we need to figure out what early pharma pipelines are focusing on. We really need to reprioritize, as I said before, the probes that we want to develop. I think the way in which we do that, I guess, is across many different ways. It's interesting. I'd be interested in thoughts from others on what the most effective ways of doing that. We can do it we can do specific calls, for example, cross-company, cross-industry collaborations in a specific area. I think it's very hard when you open it up to everything that everybody wants, because I think the wish list becomes about 20 or 30 new radio tracers. So honestly, Terry, it's great. I don't know how to answer that question. I think we just do more of that interfacing. Maybe we have to do it on a company by company basis in some ways, What I've been really encouraged by is my engagement with immunology colleagues that have never used PET before. And the insights they're giving into directing what we could do with PET has been phenomenal. So it's bringing those worlds together. And again, it can be done broad in a targeted way, but I think we need to invest more time. We need to spend more time in pharma articulating those challenges back to academia and then finding out the right ways to fund those development pieces.
Thanks, Phil. Thanks for the questions. So we have a couple more questions to go. I think we might go 1 or 2 minutes long on the timeline of the webinar, just to address all the questions that we have currently. There's one in the chat as well, which we'll come to. So another question from Phil Blower from KCL, which is again around funding, and this could help academics understand how to position their funding requests for some of the areas they're looking to research potentially, and whether pharma might expect to fund basic mechanistic studies as well as outcome measurements, or whether these basic mechanistic studies might be left towards academics to seek funding or seek other non-commercial routes to drive.
I mean, I think the answer, I like to say the answer is yes, but it's not always easy to do. What I would suggest is that I think the answer is yes, you could do that if we can see that there's a subsequent track to take it towards, impact. I think where, as you said right at the beginning, pharma carries so much risk in terms of drug development. There's a reluctance to invest in early technologies that are not drug development directly, like PET. So I think it depends on timing. If we can see something getting to the clinic within 5 years, for example, then those early mechanistic studies seem very reasonable for us to fund. Absolutely. I think I just find it quite hard when we're going beyond that sort of time frame into the longer term. When is the impact going to be? Is it going to be 3 years, 5 years, 10 years? So I think it's just a matter of timing, but we should be funding them. I think it's just creating the right value case. And I noticed there was a question about value case there too. That's a really important point because I think we need help in articulating the value opportunities of these methods.
And we'll come to Luigi's question next. So Luigi from Cambridge. So I'll paraphrase Luigi's question. So they typically find that there is a need to propose projects to industry, and there's a rather low success rate in the uptake of these projects. There's a sense that industry are not always in agreement with this approach and want to go straight into assessing tolerability and efficacy without the use of certain information. The question is how industry could maybe be brought on board with some of the projects that are proposed.
I'd say the answer to that question, and it's a challenge I have as a sort of PET advocate within industry, and seeing the pipelines that we have and the tremendous opportunities, because I engage with this great network and I see the opportunity. I think the answer is to try and work as early as possible with drug pipelines, figuring out, even at the preclinical stage, what the opportunity is, start to build the value case, build the technical opportunity, the confidence in the measurements. So I think it's about getting engaged as early as possible. Where I think, Luigi, the problem often I see is that things are proposed relatively late on in a program, and you've got this drug development machine around a compound, and it's really hard then to start to put programs related to those assets in. But if we start early, I think it's building the case as the drug development program progresses. I absolutely get the point. I think there are probably better ways we can do it from our side of things, to get more support for those types of activities.
So thanks, Phil. So the final question that we've had in the Q&A is around, it's from Bal Sanghira, who I believe is from the Barts in London. And I can see that Heather Williams has replied to the question, and I'll make sure Bal and Heather are connected after the webinar, rather than addressing that question today. But we just have one more to touch on just before we close, which is in the chat. It comes from Luke Bird from PSICRA. So do you think long-axis field of view PET will catalyze a shift in PET imaging into other clinical pathways for oncology, such as EBRT, where Abscopal.
Scopal.
Thank you. Immunoreactions are core components of tumor response post-irradiation. Currently, PET's only used before and after treatment, but not during.
I think yes is the answer, definitely. We're starting to see examples of that. It's a really interesting direction.
Okay, great. Any closing comments, Phil, Natasha?
No, thank you. Please, reach out to either of us. I think Natasha and I, different companies, we work together on different cross industry collaborations. I think that is the way to go. I think we've talked about that in a number of areas today. So yeah, reach out to us with any ideas how we can work together.
Yeah, thank you all for attending.
Thank you. So thank you for joining us today for this webinar from the National Total Body PET Imaging Platform series, looking at the current state and the future perspective of long-axis field of view PET imaging. I'd like to thank Phil and Natasha for their expert presentations and insight today. Their presentations have highlighted the growing importance of advanced imaging technologies and applying these to clinical research and development. I'd like to highlight that the recording of this webinar and future NPIP webinars will be communicated through the NPIP website, and I'd like to thank you all for attending today. Thank you very much.